Okay, so now we're going to talk about glycosylation of proteins, which is a translational modification of proteins. And we'll also talk about how proteins get targeted to areas in the cell. And so, again, we're going to talk about glycosylation. There's N-glycosylation, which is what we're going to primarily be targeting. And there's also O-glycosylation, which we're not going to talk too much of here, but we'll talk in the future. And then we'll describe how proteins are going to get targeted. We know how they get targeted to certain regions, such as your lysosomes, and how they get targeted to the nucleus. But in general, we are going to have a signal sequence. And the signal sequence is going to be directing the proteins to an appropriate location of a cell. So, for example, proteins that are going to be targeted to the mitochondria or the endoplasmic reticulum are going to have a signal sequence at their amino terminus. So protein targeting is going to be a very complex process. You're going to have proteins that are going to be secreted or integrated into the plasma membrane or targeted to lysosomes, for example. And these are going to share steps that begin in the endoplasmic reticulum. Endoplasmic reticulum is the site of many post-translational modifications. And so your proteins that are destined, again, for the mitochondria or the nucleus or chloroplast are going to have different mechanisms. Destined for the cytosol remain where they are synthesized. So here is a diagram that shows some targeting of your protein to the endoplasmic reticulum. And this looks very busy, but it's actually pretty straightforward. It's just walking you through steps. So again, you have your messenger RNA, which has that lovely 5' cap. And then your messenger RNA is going to start to be translated into a protein. In the process of being translated into a protein, you all of a sudden come across what's called a signal sequence. And the signal sequence kind of stops the translation process. And it says, this protein is not meant for the cytosol, so we're going to have to take this ribosome and incorporate it into the endoplasmic reticulum. And it does this through a signal recognition particle, the SRP. And the signal recognition particle is going to guide this ribosome with this particular messenger RNA to the endoplasmic reticulum. And it's within the endoplasmic reticulum that you're going to then start to build the protein, and it goes into the lumen of the ER. And eventually it'll be released into the lumen of the ER. Now this growing protein, oligosaccharide, is going to move into the ER. You're going to have a special protein, dolichophosphate, that is going to be important in N-glycosylation of proteins. So this is at the amino end of the proteins. And so again, dolichophosphate is going to be associated with the actual endoplasmic reticulum membrane. And dolichophosphate is going to be important for adding carbohydrates, mannose, glucose, to a growing protein. So in the ER lumen, the first step is going to be in the construction of the N-linked oligosaccharide, the N-linked glycoprotein. And so dolichophosphate can only add carbohydrates to the N-linked, and it's always going to be an asparagine end. So in this, you're going to have the core of the protein, and the oligosaccharide is going to be transferred from that dough-licophosphate to the asparagine residue of the protein. So all of those sugars that dough-licophosphate was building up will be transferred to that residue. And so now you have these sugars, glycosylation of the actual protein. Now the dough-licophosphate is then going to be translocated back to the cytosolic face of the ER, and then you're going to have the phosphate removed, and the dough-licophosphate is going to be regenerated and start building more sugars on its molecule. Now proteins can be moved into different locations in the cell. One of them that is most characterized is lysosomal proteins. These are going to be, as they go through the Golgi, phosphorylated on mannose residues. And this phosphorylation on mannose residues is going to signal that these proteins need to go to the lysosome. There are also going to be a series of steps in moving proteins into the nucleus. As you can imagine, the nucleus has the pores. So the proteins have to be able to get through the pores and into the nucleus. And this is going to occur through a series of steps. So you're going to have a nuclear localization sequence. And similar to the sequence that guided a protein to the endoplasmic reticulum, this sequence is going to pick up a nuclear protein made in the cytosol and bring it into the nucleus. So you have your protein with some sort of signal, and this is going to get bound by your nuclear localization sequence. Now, what's important about this nuclear localization sequence is it's never going to be removed, and it can be located anywhere along the primary peptide sequence. It doesn't have to be in the amino end or carboxy end. It can be in the middle of the actual protein, so that doesn't matter. The nuclear localization sequence is going to have several basic amino acids, such as arginine and lysine. It is going to be bound by another protein called importin. And this is an alpha and a beta. And then this is going to bind to the nuclear pore. And it's going to be able to get into the nucleus. Now, a lot of cells are going to or a lot of proteins are also going to need to be imported into the cells. So many of the proteins needed for cells come from the outside in. And so we're going to have to pick up hormones. We're going to have to pick up low-density lipoproteins, etc. So proteins can be taken up by receptor-mediated endocytosis. You can also have them taken up by clathrin and caveolin, dependent endocytosis. So endo means to come in. Exo would mean to go out. So in this case, we're looking at things that are coming in. And so we have clathrin-dependent, caveolin-dependent, and clathrin and caveolin independent pathways that can be used for bringing things inside of the cell. And all of these processes are going to bring them into what we call an endosome. And an endosome is a compartment that is you can think of it as like a holding spot. So the cell has to determine what's in this endosome. Do we really need it, or is it viral, or is it a bacteria? Is it something that we need to degrade, get rid of? And so all of that occurs in those endosomal compartments. All right, so in summary, we do have proteins that are going to be made. They are going to be needed in certain areas of the cell, or they're going to be released into the exterior of the cell. The peptide sequences that are going to help to direct, especially to our glycosylation for our ER, are going to be at the amino terminus. We're also going to have some peptides at the amino terminus direct to the mitochondria or to be excreted. Signal sequence are going to be recognized by some particles. We have the one for the ER, the signal recognition particle. We are going to have N-glycosylation in the ER. We're going to have O-glycosylation, and this occurs in the Golgi. We are also going to have mannose residues that are going to send your proteins to lysosomes. We can also have things get released from the cell. We can have things get taken up by the cell through those clathrin-dependent, caveolin-dependent, or independent pathways.